Okay, so welcome everyone to this week's Organismal seminar. And I have the great pleasure to introduce Ophélie Ronce. She's currently a professor at Montpelier at CNRS in France. Prior to that, she did a postdoc with Mark Kirkpatrick at the University of Texas at Austin. And prior to that, she was at the Pierre and Marie Curie University, which is 6 in Paris, working with Jean Colbert on a master's degree. And she also did her undergraduate work there. So that's the boring academic details out of the way. Ophélie works on a bunch of cool things that she'll talk to us about. Adaptation, gene flow, evolution, and dispersal. But also, I've had fun interactions with Ophélie, and I was trying to count these. This is the 4th time we've met. The first time was over whale meat in Trondheim, where we were both opponents for different defenses, and they took us out for dinner, and they took great pleasure in feeding us whale meat. I had to call home and make sure it was okay with my wife coming to eat whale meat. And then the next time was on a boat for a conference in Leiden, where we went through the canals and argued about things, range limits and things like that. The 3rd time was in Seattle just recently, where it was a symposium on evolution and plasticity, which involved a late night drinking where we all stumbled home and we were all surprised to see each other the next morning. And so I'm hoping that we can reprise those kind of craziness tonight. In particular, one thing that's shared with all these experiences I've had hanging out with Ophélie is that she has a wonderful argumentative nature. And so we've had many good discussions and disagreements, and usually people can agree to disagree, but here sometimes we disagree that we disagree and agree that we agree. So all kinds of things are possible, so please take an opportunity to have a chat with her after her seminar, because there will be a reception, at which I'm told we only have Quebec cheese, right? Because we have someone from France, and so there's no point in having French cheese. You can try the different French cheeses. Real Pelle will be here, by far the best French cheese. So today, Ophélie will talk to us about models of adaptation to climate change for complex life cycles.
Okay, thank you very much, Andrew. So I actually gave a talk here 15 years ago where I was doing my postdoc. It's not back in Texas, so I'm quite happy to be back here. So I put out some orange slides to bring a bit of spring colors to my show. And so I'm going to talk about different models of adaptation to climate change, trying to introduce in those models some feature of populations that have been a bit ignored in this context, which is the fact that population contains often individuals in different stages with different demographic properties and contribution to population growth. And my current research is investigating how this might affect the dynamics of adaptation to changing environment, including climate change. So I tried to cover all those 4 points during this talk. It's going to be a bit a challenge. So I'm going to talk about work that I did not do, but just to compare to what I've been doing, about ways that people have attempted to formalize the adaptive challenges associated with climate change and the demographic consequences of it. Then I'm going to talk about what is going on in such heterogeneous population where you've got different individuals with different ages and different contribution to population growth, and suggest that climate change could result in faster aging of those populations, and I will explain to you why. And then the 3rd point, I tried to see how climate change might affect trade-offs between different life history traits and change life history strategies in adaptive or maladaptive ways. And finally, I will tell you about current attempts that I'm doing in collaboration with other workers in Montpellier to try to parameterize those very theoretical and abstract models with insight from mechanistic models of tree ecophysiology to try to better qualify and quantify those adaptive challenges associated with future climates. So let's talk about what was done before and about climate change. So it's not completely new and exciting, but so what we know is that climate warming is favoring probably very different phenotypes from what we found in population currently. There's been different attempts to describe that and try to predict that. So this is one famous study on red tits, where the people have studied laying date of the birds, and there's very strong selection pressure to make the laying date match the peak of food abundance in those populations. When the peak of food abundance is related to the development of insects and caterpillars on which the tits feed on. So with different scenarios of climate warming through years, with fast warming or slow warming through years, those authors have predicted what would be here with the empty symbols, the optimal date of laying that would maximize chick survival. And so you see that depending on the scenario, you've got a decrease in optimal laying date that could be fast or slow depending on the rate of temperature warming. And in the field symbol here represent what the birds are predicted to do based on current reaction on to temperature in terms of phenotypic plasticity. So what you see is that in all scenarios, you've got an increasing lag with time between what birds should do in terms of phenotype and what they're able to do with their current reaction on their current phenotypic plasticity. So this means that in those populations, you've got increasingly strong selection for genotypes with earlier laying dates and different reaction laws. So we've got selection for different phenotypes. Do we see change in phenotypes going on in population that can relate to climate change currently? So yes, there are not that many evidence, but a little bit. So this is one very nice study as well that was done on some brassica species, where they use resurrection experiments where they grew seeds collected in 1997 and seeds collected in 2004 together in the same common garden in a dry site, in a dry site and wet site. And what you see is that when you grow together the plants and look at their date of first flowering, there's been a shift in date of flowering that is moderate in this site but pretty big in this site. So you would see fast evolution of phenotypic traits in those populations. But how is this fast evolution fast enough in terms of meeting the challenges of climate change? So this is to answer this question that some modeling has been developed to understand when fast evolution is fast enough or not fast enough. So this is actually dating back from the early 90s and from a series of models, in particular this first one by Lynch and collaborators. And the simple idea behind this model is that you assume that the contribution of every individual in the population to the growth of this population, so their Malthusian fitness, depends on their phenotypic trait, which could be laying date, for instance. And what you assume is that there's some relationship between this contribution to growth of individuals and their phenotypes with this kind of bell-shaped curve. You've got some optimal phenotypes that maximizes fitness. And so, and what climate change or environmental change is doing is that before the change, you had some value of the optimal phenotype and some shape of the function describing selection in the population. And with climate change, you're shifting this distribution in some direction. So the optimal phenotypes are changing, often are assumed to change linearly through time with some speed k. So this is what's happening when you look at different individuals in the population. Now, if you talk about population demography, what you want to know is what is the mean population growth rate. So to know the mean population growth rate, you have to integrate across the whole phenotypic distribution the value of those individual contribution to growth rate of each individual. So it means that for those models assume that you've got some Gaussian distribution of phenotypes in the population, and from there you can compute the mean population growth rate. So for instance, this is a distribution of individuals that were adapted to this former blue environment, and this new environment will have some mean fitness that is determined both by the variance of phenotypic trait in the population and how far they lag from the optimal phenotype. So more precisely, in mathematical terms, you can write this mean population growth rate as a function of different demographic or fitness loads. So here's the mean population growth rate This is the population growth rate that you would have in that population, in that environment, if every individual in that environment would have the perfect phenotype matching the optimal phenotype in that environment. So that's the maximal population growth rate. So this effective mean population growth rate is going to be smaller than this maximal growth rate, in particular because not all individuals in the population have the same phenotype. So even if you have the mean phenotype matching the optimal phenotype, you will have some phenotypic variance in this population, which means that some individuals will deviate from this optimal fitness. And so you have what is called the variance load, which is depressing the maximal population growth rate when the phenotypic variance increases in the population, scaled by some selection intensity measure. And then there's a second load that is useful to distinguish, which was called the evolutionary load or lag load, depending on papers that you will find. And this load is describing how your mean population growth rate is declining compared to this maximal population growth rate when the mean phenotype in the population is deviating from the optimal phenotype. So it represents some lag between what the population evolved and where it should be in terms. So it's also related to selection gradients in the population. So what's going on is that imagine you had a population with some size initially and some phenotype here, and then you start changing the environment through time. So the optimal phenotype is changing linearly through time. And what's going on is at the beginning, the mean phenotype is not changing much. And then it's the lag between the optimal phenotype and the mean phenotype increases. Selection becomes stronger on the phenotype, then start to respond to this selection, start to evolve and track the optimal phenotype, but with some lag that will become constant and stabilized. It means that's why I put the picture of Alice in Wonderland at the beginning. The population is evolving all the time, running after the environment, but always being late. So there's always a lag between where you should be and where you are, because you can't evolve as fast as the environment will change. You will always be behind it. So it means that, and so the population size will decline as this lag increases through time, and then it will also stabilize. So this is the population growth rate. So population growth rate will decline through time as the lag increases, and will stabilize when this lag becomes constant. And if this lag is too large, so that it results at this dynamic equilibrium to a population growth rate which is below zero, which is the case here, then you will predict certain extinction of the population. So this model is used just to say when the population is doomed, basically, that when it can evolve to track the population not fast enough so that it will accumulate a demographic load that will not allow to maintain a positive growth rate. And if at equilibrium you evolve a lag load, a lag that is smaller than this critical lag, then you could persist, but you're not guaranteed to do so. You have to take into account more stochastic factors. So what is nice about those simple models is that you can have a very simple expression about this lag at equilibrium. So this lag scaled here by the intensity of selection will be larger. So this is the distance between the optimal phenotype and the mean phenotype at any point in time, which will be constant. This lag will be larger as the speed of climate change is greater, so it's quite intuitive. If the climate is changing fast, then you are lagging behind more. And what is also intuitive is this lag will be larger if the genetic variance is smaller, which means that you evolve more slowly, you respond more slowly to selection, so you will lag behind more as the genetic variance is smaller. And so from this, you can define critical speed of climate change such that this lag will be too large and you will evolve a negative population rate. So you can try to predict which kind of climate change, or the speed of climate change that you can persist under or not. So that's for previous theory. Now, the question I was interested in is that many populations actually house individuals in very different stages, such as tadpoles and terrestrial stages in the life cycle of old or young individuals. And there's evidence that climate change may have differential effect on individuals in different stages. For instance, the climate change pressures on tadpoles and terrestrial adult stage might be quite different. You could have one stage that's more affected by climate change than another. You could also have different traits or genes that are contributing to adaptation at different stages. So all stages could be affected by climate change, but it's not the same genes that would respond to selection to improve performance in the different stages. And finally, a different scenario, which is kind of the opposite of this one, that the same trait might be expressed by the different stages in the life cycle, but it could be under different selection pressures in the different stages. So it might be good under a warmer climate to increase some trait when you're juvenile, but bad when you're an adult. So just an illustration of that. In the lab, we also are conducting experiments on spider mites, looking at evolution, adaptation to new hosts. And so this is not climate change, but it's an illustration of adapting to a new stressful environment. So those lines evolved for a number of generations on cucumber. And so this is the ancestral line. And what we did is that we had lines evolving on pepper and lines evolving on tomato, which are those red dots and yellow dots. And what we have here is assessment of juvenile survival of the ancestral line on tomato and on pepper, and on the tomato line on tomato and pepper, and the pepper line on tomato and pepper. So what you see is that after only 15 generations of evolution on the new host, the tomato line have evolved much better juvenile survival than the ancestral lines, and the pepper lines have evolved better juvenile survival than the ancestral lines on pepper. So it's nice, you see ongoing adaptation, but if you look at the different life history traits on the very same lines, which is adult survival, which was also genetically variable to start with on both new hosts and the population, we see no evidence of improvement in adult survival in the tomato lines or the pepper lines. So adult survival did not improve significantly in any of those lines, but another trait expressed at a different stage in the life cycle did improve. So we see that the dynamics of adaptation vary with life history traits, but we don't have prediction about which trait will adapt faster, and we don't know how climate change or different environments will affect trade-offs between those traits and whether they could result shifts in life histories. So that's where I wanted to produce statistical prediction to get a better understanding of those questions. So it's kind of surprising that we didn't address that, because we actually have very good sophisticated modeling toolkits to look at adaptation in age or stage structure population that was developed by others than me. So starting with the work of Russ Landy in the 80s, and then more recently collaborators in Norway like Steinar Engen and Gabriel Setter developed a lot of nice quantitative genetics models in age and stage structure population. There's also this very nice paper, if you're interested, by Barton, Belfield, and collaborators for stage structure population. So now you're looking at population that is a bit different from what we were modeling before. So in that population, you've got different intervals, for instance, here with different ages that have different fecundity, fx, or different survival from age x to x plus 1. We've got juveniles that do not reproduce and adults that reproduce, and we're modeling a more complex object, but we do have tools to understand evolution in those. So what are those tools saying? So you can predict under weak selection the rate of evolution in a mean trait value in such an age structured population. And so this is a result from this paper and Engen collaborates paper. So the rate of change in mean phenotype in one time step will increase with the amount of genetic variance in newborn. So this is quite equivalent to what we've seen before. And then instead of having a selection gradient that is describing just the contribution of individuals to population growth rate, you can decompose that in terms of the effect of variation of the trait on mortality at each age and fecundity at each age. Each of those being weighted by those weights here that are called demographic elasticities, which are classic measures in demography. So what are those? So it means that you can look at the effect of trait variation on each of these life history traits. And those demographic elasticities are well-known quantities that just describe the effect of perturbation in life history transition in the life cycle on the population growth rate. So, and those have well-known expression in age-structured populations. So what you do then, you do something very similar to what I presented with the Lynch model. Instead of mapping mean fitness with phenotype, you're going to look at the specific effect of variation in phenotype on each of these life history parameters, like age-specific survival and age-specific fecundity, and you can similarly define the strengths of selection and the optimal phenotype, maximizing each of these life history traits. And you can assume, as we did before, that optimal phenotypes for one trait or many change to time linearly with some speed k that could be the same or different depending on life history traits. So now we can define again some kind of genetic loads, but instead of looking at it through the total population growth rate, we can see how those maladaptation and genetic loads affect each life history trait. So we could look at mortality rate at age x, which is a log of the survival, mean survival at age x. And this will depend, this mortality will depend on some basic mortality, which is increased again by a term which depends on the phenotypic variance in the population. So this is the equivalent of the variance load, but expressed at the level of age-specific mortality. And then again, you recognize the term which corresponds to the lag load, which is how far the mean phenotype in the population is lagging behind the optimal phenotype that maximizes that life history trait, not the population growth rate, but just this trait. So if you do that, and you keep on doing a very artificial exercise that is helpful to understand what might be going on, and you assume a trait that would affect only mortality at age x. So it's a bit artificial because you assume that this trait has no effect before age x, no effect after age x. It's just affecting the transition between age x and x plus 1. But we'll see that once you computed that, you can combine those to have more realistic understanding of what is going on. And so if you imagine such a trait and you compute what would be the lag between the mean phenotype in the population and the optimal phenotype that maximizes this survival quality at age x, we've got an expression which is quite similar to the one that we had before. So if the mean phenotype evolves to track the shifting optimum with a constant lag as before, then this lag is going to increase with the rate of environmental change, no surprise. It's going to decrease with increasing genetic variance in juveniles. It's okay. But then it's changing with this term, which was this elasticity term, with describe the effect of perturbation in age-specific survival on the population growth rate. So this measure of elasticity is actually measuring the strength of selection on age-specific mortality. And this quantity was used in life history theory to understand the evolution of aging. So we can compute that, and actually Bill Hamilton, I think, was the first to do that in his 1966 paper. If you compute the strength of selection on age-specific mortality, you find that you've got this expression. It might look ugly, but I will explain it to you. So this is actually the residual reproductive value that you will have if you survive to age x plus 1, scaled by the duration time in the population. It's actually the sum of the probability that you're going to survive to age y, the fecundity that you will have if you survive to age y, just with some weighting factors that depend on population growth. This is just summing your expected future contribution to population growth if you reach this age x plus 1. So actually this quantity is always decreasing with increasing age x, because it's a sum, and so if you stop this sum later, you would just have the same terms minus some terms. So it means that if a gene is expressed late in the life cycle, then the contribution to future reproductive events will be just less than if you have a gene that is expressed later. So this quantity is always decreasing with age, and this was used as a theoretical argument by Bill Hamilton to try to explain why senescence is such almost universal phenomenon in living organisms, from yeast to complicated things such as humans. So for instance, this is just one example of computing this elasticity with demographic data from human populations. And so you see the strength of selection as a function of age. And what this predicts is that the strength of selection is starting to decline as soon as you reach the mean age of foster production in the population. Then it declines at every reproductive event in the population until you reach the last age of reproduction in the population where selection intensity is just zero, acting on mortality. So this was called somehow sometimes the wall of death, because any mutation that would have effects expressed only after this age will have no selection to oppose its spread in the population. It will be just neutral. So what does it predict in terms of adaptation to climate change? It predicts that adaptation to climate change depends on traits that have a differential expression with age. You would expect larger adaptation lags for traits that are expressed late in life, because this quantity is decreasing with age, so the lag should increase with age. But this is maybe too simple an argument, because there's another quantity here that might depend on the age of expression of the genes, which is the amount of genetic variance you expect for those traits in the population. If a trait is expressed only late in life because it's gonna be subject to weak selection against it, you can also expect to have more genetic diversity for this trait because it's not as strongly selected as a trait that will be expressed early in life. So you've got this quantity which is decreasing with age, but this quantity is supposed to increase with age. So if you look at the genetic variance, you would expect actually lower adaptation lag for traits expressed late in life because they have more genetic diversity to respond to new selection pressures. So actually work that I'm doing in collaboration with mathematicians at the moment that are based in Lyon shows that this product, actually you can show that it's always decreasing with age. So the effect of the declining strength of selection is dominating the effect of larger genetic variance for those traits. So actually what this expression predicts is that you do, under this very simple assumption, expect larger adaptation lag for traits that are expressed late in life rather than early in life. So just to test this analytical prediction and relax some assumptions that we made there, we look at this very simple toy life cycle with 4 age classes, one age class that does not reproduce, like a juvenile age class, and then 3 adult age classes. And we've got 3 phenotypic traits, each affecting different survival rates, and we can have those traits evolving, be genetically independent or have some genetic correlation between those traits that could be negative or positive. So it allows us to relax some assumptions. And then we let the genetic variance evolve in those simulations using some infinite model of inheritance. So we assume many loci of small effect affecting each of the traits with some correlation on it. So what does it predict? So this is the rate of mortality as a function of age for this hypothetical species undergoing no climate change, so that's a k equals 0 case. So we've got slight increase in mortality with age in this population in a constant environment. But if the environment is changing faster and faster, you see that mortality is increasing with age faster and faster. So what we see in the simulation is that we've got much faster aging under rapid climate change than under any constant environment. And if we try to understand better what is going on there, so here you've got through time the mortality in the different age classes with slow climate change. So this climate change is initiated here. So initially, even in a constant environment, you see that mortality is increasing with age. So we've got high mortality at age 3 and initially, while we did not build that in the model from the start. They had initially the same basic mortality rates, but you see evolution of indeed senescence in the model. But what's going on is that if you start to change the climate, the mortality in the late age class is increasing much faster than in other age classes, but it's increasing only slightly. And this is due to the fact that if you look at those loads on mortality that I described to you before, if you look at young age classes, so what this graph shows is through time the mortality rate, and part of the mortality rate, which is the basic mortality rate here in the dashed line, and the realized mortality rate here is entirely explained in the young age classes by the variance load. It's just explained because there's genetic and phenotypic variation in that age class, for genes expressed in that age class that is increasing the mortality rate. In the old age classes, what you see is that you can also decompose the mortality rate in terms of basic mortality, extra mortality due to variance load, and extra mortality due to lag load. And you see a higher variance load in older age classes, which is consistent with the fact that there's more genetic variance maintained for traits that are expressed late in life. So that's what explains most of the aging processes in this model. But what you see here that we didn't have here is that for old age classes, you also have some part of the mortality which is explained by the fact that the mean phenotype for those traits that are expressed late are departing from the optimal phenotype. So there's an evolutionary lag load on top of it. So if you have fast climate change, what we see is a bit different. So it's the same story. You've got higher mortality even because of higher variance load, even in constant environment. But if you start to change environment very fast, you see that the mortality in the last age classes is not stabilizing. It's increasing and diverging all the time. So this is something new. And so here you can see that this divergence of mortality is due to a divergence of the lag load. The variance load is quite stable actually, and is what you expect in a constant environment. So again, with those mathematician colleagues, we could show that there's actually another critical speed of climate change, which is lower than the rate of climate change that will cause the entire population extinction. But for this critical rate of climate change, you will have lag load diverging after age x, which means that the survival probability after age x will